So today we're going to be talking about configuring data analytics workflows for faster biomarker discovery. So when doing that, I want to talk a little bit about some of the challenges that we have in life science regarding big data, because I think that's what we're really talking about is some of the big data challenges. And the way I look at this and the way we look at it at PerkinElmer is there's really one side which is kind of the variety, the volume, the complexity of the data, things like where the data is coming from, the gigabytes or terabytes of data that's being generated by next generation sequencing equipment and other sorts of tools out there, as well as the complexity when you think about structured data versus unstructured data. So there's a lot of challenges associated with that as far as kind of what the data looks like, how the data is shaped, and what you're going to do with the data. The other problem we really see is kind of the other half of the coin, which is that most people work in isolation when working with this data. They don't really have the collaboration framework or the infrastructure to manage that today. So oftentimes when starting a biomarker project where you're looking for a particular biomarker or you're trying to discover something there, you have to find the clinical trial data that you need to support to actually conduct the analysis, aggregate all that data together, and then do your analytics on that. But all of that is kind of left out when you're done. It's put on the shelf somewhere. It's never really reused. And that infrastructure to be able to share that type of analysis with colleagues and be able to gain insights on that more quickly is kind of what we're trying to do here. So what we're trying to do overall with some of the solutions we have in place are really driving towards focusing on the high-value activities. So when I think about what goes on in translational medicine in particular, there's a lot of effort spent on collection curation, which is why this red line represents kind of the current effort that people are spending. So they spend all this time on collecting data, curating data, and they really don't have much time left over for analysis or insights on the data. And with the solutions we're offering, we're trying to shift that curve to look more like this blue line, where you can spend less time on collection and curation and then have more time for analysis and insights. And that's really the direction we're going. So with all this in mind and kind of the idea of what translational medicine is and those sorts of things, the other thing we want to be cognizant of are the various workflows that the translational scientist kind of goes through. So when you think about them starting from raw data, doing data processing, analyzing all that data, and then doing secondary and tertiary analyses after that, and then on top of that, then taking those secondary analytics and then reviewing them even further, processing them further, comparing them with other clinical data that's out there, and doing other integrated analytics. So enabling that entire workflow is something that typically people have to do, on their desktops, and they have to get data and do it all themselves because there isn't really a system out there that helps them through the entire process with this workflow. So I view that as one of the major problems in the industry today. The other thing to think about too is how we view translational medicine overall, and what it really boils down to is what I was talking about earlier, which is biomarker discovery. Just to clarify what we mean by that, when looking at a patient population and looking at all the various measurements and all the data that might be collected off that patient population, we then want to analyze that data to find out what are the differences among these patients so that we can get the right drug to the right patient at the right time. So once we find all those differences and we can categorize those as those various biomarkers, now when a patient presents themselves, you can take those same measurements and then you can identify where they fit within that stratified population and provide them with the right targeted therapy and a precise diagnosis so they can actually be treated more effectively for their particular ailment. That's the entire workflow we're really trying to enable here. So when you think about that workflow and all the different items that I just mentioned, there's a lot that's involved that you kind of need to do that. So at the bottom layer here, you need to be able to store data. You need to be able to bring data in from your various sources. I mentioned clinical trial data, but there's also other sorts of data. You have real-world evidence data, electronic health records, imaging, clinical trials. And all this could be in standards like CDISC. It could also be from public sources such as GEO, or if you've got an Odyssey framework in your existing environment, or even Transmart, something like that. You've got to be able to take all of that data together so then you can curate and normalize that data and actually harmonize it so you can do some sort of search across it and identify the patients.